So good morning. Thank you for the invitation. I'm very happy to be here. So yes, I come from Paris, and I will give a tutorial on optimal transport. And hopefully, by the end of the talk, you will understand what is this kind of interpolations between distributions of mass and space. So before that, let's start with the beginning. More than 200 years ago, Monge, a French mathematician, posed the problem of transporting a pile of dirt from one configuration to another. He wanted to do it in the most efficient way. And so he started to think about this problem. And what we will keep from this problem is that we have two distributions of mass in a space. We'll call them mu and nu. And we'll find the most efficient way to transport one distribution to the other. So you may think, why mathematicians, 200 years after this, they still think about this problem? And one of the reasons is that it has actually a very strong modelization power, this notion of distribution of mass. It can be used to describe probability distribution in a space. It can be used to describe a mixture of weighted particles. And actually, I won't go into all these applications, but you can think of this as images. Optimal transport has been used to study the early universe, where here each particle of mass would be a cluster of galaxies. It has been used to process histograms of colors. It has been used to study crowd motions, and everything basically which can be represented as distributions of mass in a space can be dealt with optimal transport. And I will show you that optimal transport gives a nice structure between these kind of objects. So the plan of my talk, I will first give, in the first part, I will lay the models and the basic theory of optimal transport. I will try to stay at the high level. And in the second part, I will talk about a new class of numerical methods which has emerged in the recent years, which allows to solve this kind of problem quite rapidly and to reach new applications. So in the first part, let us start with the mathematical formalization of this problem, going from pile of dirt to mathematical objects. So first, what we need to do is to decide where our mass will lie. So we will choose a space that we will call X, where we will have our particles of mass that will live. And we need to choose a cost function which tells us what is the effort of moving one unit of mass from one point to the other in this space. So that's the cost function. It can take a real value, or it can be also infinite if I want to prevent transport to happen between 2 points. And then I need to choose two probability measures, mu and nu. Why do I call them probability measures? Because these are distributions of mass which have a unit mass. Because if I want to transport one to the other, of course, the mass needs to be preserved, so they are normalized. That's an important point. I will come back to it later.
And feel free to interrupt, as always.
Of course.
Especially in the primers, but always. Is there any questions?
For example,
if any of this is unclear, you're going to be lost for the rest of the hour. So
a reasonable question is,
yeah,
probability measure versus probability distribution.
Okay, so I will use indistinguishably all these words. Distributions of mass, measures, these are the same mathematical object here. That's important to keep in mind. We just think of mass which is distributed on a space with some configuration. And the important fact is that all the particles of mass are indistinguishable. So yeah, we just, and when we call it a probability, it means that the mass sums to 1. So now I will have to decide for a way to describe what is a transport plan between two measures using a mathematical object. And this object, I will call it gamma. And it tells me how much mass in my plan of transport is moved from the point X to the point Y, okay? So gamma of day X, day Y, it tells me how much mass is moved from a little volume around X to Y. And with such a function, I can describe a way to move all the mass from mu to the mass of nu. And then the cost associated to this whole displacement thing will be computed by writing the integral over the whole set of pairs of points on my space of the cost of associating this pair of points times the amount of mass that I will transport between these 2 points. So this is this integral. And solving the optimal transport problems problem is just to minimize over all possible transportation plan this objective. I will call it objective. It's a function that I want to minimize. But of course, there are constraints on this gamma. It needs to be something which describes a plan. Yeah?
So some of the dirt sitting above X might go to one Y, and some of the dirt sitting above X might go to somewhere else?
Yeah. So in this formulation, we do not force mass from one point to go in only one other point. It can split, because with this kind of description of transport plan, I can split mass. The mass at some point can also come from several points. It's not implied that my transportation will be given by a map. So I want to talk a little bit about the constraint that I have on this object that is called gamma, which describes a transport plan. So we call it a coupling. What is a coupling? It's a distribution of mass on the pairs of points, so this object gamma, as I described before, which actually describes the way to transport the mass from mu to the mass of nu. And it can be thought of as this way. For instance, if here I have distributions on a one-line segment, mu in red and nu in blue, a coupling which is admissible is, for instance, this gamma. Here, it describes a way to transport the mass from mu to nu. How can you think of this? You start from a point in nu, you look at where the mass is sent. This gamma will describe where the mass from this point is sent. So here in this description, for instance, the mass is scattered over the whole other distributions. Okay? Here it's another example with a very different behavior. When you look at here, you go where you see where the mass is sent, and there's actually only one point. So it means, we call it a deterministic coupling. It's a way to distribute mass from mu to nu, but that's the case that Alex described, where you don't split mass. Where you start from one point, you will go to another point. And the optimal coupling will tend to look like this. I will talk about it later, yeah?
So in that case, there's just a simple mapping from the points of the red space to the points of the blue space.
Exactly, yeah. In the other case, what we call deterministic coupling, it's a simple map. And in that case, this one is called the product coupling, it's just a name. It can be think of as a stochastic map. When I'm in some point, I don't know exactly where I will go. The mass will be scattered in the directions. So in terms of mathematical notation, we say just that gamma is a mass on the product space, because here you see that it's we take for one-dimensional measures, the mass of the gamma will be distributed on the plane, and its first projection is mu, and its second projection is nu. You can think of it graphically that when you project the mass, here you get nu, and the second one you get nu.
So just to say it again, this is the sort of a key definition.
Yeah.
You're looking at measures on the product whose marginals, the marginal distributions on each of the two sides are the measures that you're trying to couple.
Yeah.
Right,
so here is another way to think of those couplings. It's just this, another representation to help you think about these objects. Here, my two distributions of mass are sum of weighted points in the 2D plane. And here it's an example of coupling which scatters mass. When I am at a point on the red distribution, which is my first distribution, I can be sent to any other direction to the other distribution in blue. And here it's another kind of coupling where, which is optimal actually for some choice of cost on the plane. So here, the thickness of the lines represent the amount of mass that is transferred between 2 points. So that's for the concept of coupling. And the fact that optimal transport is formalized using the concept of coupling has been introduced by a Russian mathematician, Kantorovich. And he actually showed that that was the good way to see the problem. Why? Because every time we have an optimal transport problem, there will be a solution. You will find a coupling that will solve the problem and that will reach the minimum. So here, I wrote big formula, but I will explain what it means. This is the so-called Kantorovich duality theorem. It gives you another way to think of optimal transport. So the first line, we have seen it. It's just the minimization problem over the set of couplings gamma with the constraint that gamma actually transfers the mass from mu to nu. And the second line, it's called the dual problem, and I will give an interpretation of what it means. It's a problem in terms of prices. So let's assume that I'm running a company, and I have a set of factories which produce goods, and I have a set of shops which will sell these goods, and I want to transfer my goods from the factories to the shops. So if I know the amount of goods which are produced in each factory and the amount of goods which are sold at each shop, then what I will have to solve, assuming that the cost of transporting the goods from the factories to the shop is linear and proportional to the mass of goods or the amount of goods that are transferred, what I want to do is to solve actually an optimal transport problem. I have the cost of transporting my goods from each factory to its shop, and I want to find a plan, a way to transfer my goods from the factories to the shop that minimizes the cost that will cost me all these things. But then there is another point of view to have. I can think, maybe I will ask another company to do the job for myself. And that company, they will charge me for picking the goods from my factories, and they will charge me for dropping the goods in the shops. And the price will be proportional to the amount of goods that will take or drop. And I will ask them that the price, so phi and psi here are prices. I will ask them that the price that will charge me for picking the goods at some place and the price that they will charge me for dropping the goods at another place, it should never exceed the cost that I would have that I would have if I did the transport myself. Otherwise, I wouldn't have any interest in asking them to do the job for myself. So this is this constraint. Otherwise, if they don't satisfy this constraint, I won't ask this company to do the job. And then they will, of course, try to maximize their profit. And so their profit is just the amount of goods they pick up times the prices they will give to each good. So the price is spatially dependent. And this is the other term, which is the prices they charge for dropping the goods times the amount of goods that they drop. And Kantorovich duality theorem tells us that actually I will pay the same in both situations. The company will always find a set of prices that can make me pay as much as I did the transportation myself. So here is a representation quickly to see this problem. You have the set of factories in red and the set of shops in blue with a size proportional to the amount of goods that they produce or sell. What I will come up with if I want to minimize the transport costs is this way of transporting my goods from the factories to the shops, and that other company that I will ask to do the transportation for myself, they will come up with this set of prices. Here, the higher, the more colorful is the dot, the higher is the price they charge. So for instance, we see that the factory, which is very far away from all the shop, they will come up with a very high price for this transfer. And here, we can check that this set of prices, they satisfy the constraint. And so there is a relationship which exists between the optimal prices and the optimal plan. I don't need to go into this detail. But now let's think. So we have seen essentially the minimization property of this problem. We'll look now at the structure that optimal transport allows to give to the space of distribution on some space. So for instance, let's come back to our set of points with weights. Yeah. Okay. Yeah.
So it's not so common for a function, an arbitrary function of two variables to have the form.
Wait, sorry?
The optimality condition, right?
Oh, that's one?
X plus psi of y and c of xy.
Yeah.
I mean, that doesn't usually hold over a large area for
Yeah.
this function.
Exactly.
So
Yeah.
I guess that's some hint already that the optimal thing is concentrated on a much smaller.
Yeah. I don't know. Yeah, that's right. Yeah, exactly. This is a very specific condition, yeah. So let us look at the structure that I can give to the space of distributions. Here I'm just looking at weighted points, but it can be, think again of all the examples I've introduced, can be very much complicated objects, much more complicated objects. When I have an optimal plan to join one distribution to the other, I can define an interpolation between these two distributions. And why is it interesting? Because you can think of this interpolation between these distributions as a straight line or as a line joining two measures, two distributions, which is somehow the shortest path for going from one distribution to the other. And with this picture of mine, you can think if that's the shortest path, maybe there's a distance related to this way of looking at the distribution of mass. So that's true. Optimal transport allows to define a notion of distance on the space of distributions. Yes, there was a question?
So are you like, if you think of the thickness of the line and the length of the line, is the goal to minimize like the surface area of the line?
So it depends. That's the case if the cost that you choose is the length, the distance. In general, that's not the cost that we choose. The most useful cost, actually that's the one I wrote here, is the squared Euclidean distance. So in this case, it wouldn't hold, but if you choose here, of course, just the Euclidean distance without the square, it would be true. But the best structure, I'd say, that you obtain on the space of distributions is by choosing the squared Euclidean distance between the points, and it allows to define a notion of distance on the space of distribution. And why is it interesting? Because once you have such a nice structure, you can define interpolation, you can average between a set of distribution, you can start doing a lot of mathematical operations to deal with your data. And that's the last thing I'd like to say about the theory of optimal transport. That's the last slide on this part. Is now if we add the velocity vectors associated to these particles. That's how it looks. This is the interpolation, and I've just added the velocity vectors. So we can think, is there a way to directly characterize this interpolation in terms of a minimization problem? And that's true because each particle travels at a constant speed on a straight line. So if I compute its squared velocity, then I will obtain exactly the squared distance between the 2 points. And if I multiply this by the mass of each particle, I get from the dynamic point of view, I will get the kinetic energy of the interpolation. And if I integrate in time, I will get the integral of the squared distance times the mass that is transported. So it gives me another way to look at optimal transport, which is a dynamical point of view. We'll explain the terms of this theorem. So this quantity is just the previous cost that I've introduced, which defines the distance. Actually, there's a theorem which says that it is obtained as minimizing over the whole set of interpolations between my distributions of mass, minimizing the kinetic energy. So I'm looking for an interpolation which minimizes the kinetic energy. And this equation, it just tells me that mu-t, which is my interpolation, depending on time, it varies, and it's affected by a velocity field, v, which depends on time and space. And this velocity field, it is the one which appears in the kinetic energy here that I'd like to minimize. So these are the few facts.
It's
yeah.
to starting and ending at the right thing.
Yes, yeah, I didn't have all these notations, but mu-t, it interpolates, of course, between mu0 and mu1. So now I'd like to talk about an extension of optimal transport that will be useful in the next talk. So keep in mind that some things that I'm saying might be useful for the next talk. So in general, you want to use optimal transport because it's very useful tool to define a structure on the space of distributions, but there is this constraint that your starting and final distributions have the same mass. And sometimes you'd like to get rid of this constraint because in your data, you don't have the same mass. And even if your data have the same mass, sometimes what you want is not exactly to displace the mass, but rather the local maximum of density of your distribution. You would like to transport the modes instead of the mass. So I will show you the kind of behavior that I'm talking about. If you compute the optimal transport interpolation between these two distributions on the line, the blue and the red one, because the bumps, they don't have the same mass will be scattered. But the kind of behavior I'd like to obtain in some applications, I will study it at the same time, maybe something like that. I'd like to adapt the amount of mass which is locally available so that I can preserve the structure of my distribution. And here, what I'm transporting is rather the modes than the mass. So I will spend 2 slides on describing how to obtain such kind of models.
Just for motivation, I know it'll come up in the next slide.
Yeah.
But can you concretely, why would the second make more sense?
So it really depends on the models.
It feels to me like you're breaking all the rules, but I have no idea what you're going to do now.
Yeah. So I mean, it really depends. It really depends on your application. Every time, of course, when you come up with a model, because you have an application in mind. So.
So suppose we had, like, I don't know, single cells, and we measured them.
Oh, yeah.
Time and at a later time.
Okay, that's a good. That's a good example. I don't want to. I don't want to spoil all the talk, but yes, let's say if you have some cells and they don't have the same rate of growth, and you'd like to match your distributions of cells, which evolve with time. I'm sorry. They don't have the same rate of growth, but you'd like to match these distributions of cells. Of course, you'd like to take into account the fact that the cells have a multiplying rate, which may vary depending on their genotype. And this kind of model will account for this fact and would give a better matching, which is more natural with respect to your application.
So what replaces conservation of mass? Because that's like a very rigid constraint.
Yeah.
So what replaces that?
So in these new kinds of models, we are able to interpolate between any distribution of mass, which is just, yeah, of arbitrary mass. I can describe this kind of models. So this simulation will actually compute it with the models I will describe. Yes? Mode is a maximum, local maximum of density of a probability distribution or distribution of mass.
Like a cluster.
Yeah, it can be thought of a cluster, concentrated area where mass is concentrated. So yeah, what we need to do here, what I call the balance case is the standard optimal transport case. The way the degree of freedom we have to move some mass is just by acting a velocity field on it and displacing it. To come up with a model with unbalanced and variations of mass, we can add a degree of freedom, which would be a rate of growth, which can make the mass vary along the interpolation. And then we just copy exactly the same model as I've shown before, where we minimize the kinetic energy, but you generalize the notion of kinetic energy by also measuring the growth energy. So here, I will minimize over the whole set of interpolations between my first measure, my first distribution and the second one. I will try to find the interpolation which has the minimal kinetic and growth energy. And this modified continuity equation just describes the conservation of mass. It tells me that the variations of my interpolation are explained by a velocity field, but also by a growth rate.
So this is why you introduced the Benham-Brenier formulation.
Exactly.
Because now you have a new degree of freedom, but you don't need a new hard constraint. Instead, you have
Yeah.
to kind of regularize.
Exactly. Okay. But there is a thing, actually, that's a good introduction. We may want to generalize optimal transport directly from the original formulation that I've shown to you, where we were looking directly for a transport plan from one distribution to the other. So the issue here, we cannot deal with distributions we do not have the same mass because there is no transport plan between them. But we can maybe think of transporting between measures which are similar to mu and nu, which will have the same mass, and then I will try to make them not so different from mu and nu. And actually, the first model I've introduced admits a formulation, so it's a bit complicated, this formulation, but it can be rewritten as the kind of model I've just described, where you minimize over transport plans, a transport cost, plus some function which will measure how much the marginals of your transport plan deviate from mu and nu. And the way to measure this deviation is using the relative entropies. You don't need to know exactly what it is, just a function which is 0 if the two arguments mu and nu are equal, and which is positive otherwise. So that's all for unbalanced extension. I mention it because it's also useful for the next talk. And for this first part on theory, the main message is that optimal transport is a natural way when you have distribution on a space. You can think of it as your data, distribution of cells, or all the examples I've shown before. It's a very natural way to define a structure on the distributions on that space. So you start from the geometry on your space, and you lift it on the structure, as a structure on your distributions on that space. And it works for probabilities, but also on normalized distributions. So now we'll talk about numerical methods. Yeah?
Do you get different solutions with different relative penalties on the growth and
Yeah.
the velocity?
So there's a whole, I'd say, a whole set of possible models for this unbalanced models. So I described them in my articles, but
Is that a one-parameter family?
no. So of course you can add a parameter here. It will give you a family of different transports, but you can choose other kind of functions than the relative entropy, and it will give you other kind of behaviors, and they sometimes correspond to dynamic formulation. Not always. I've just shown the most classical model.
Sure, but then in this business about moving modes, presumably you have results that say that that is actually what happens.
Exactly. Yeah. Yeah. So it's not clear from this formulation at all.
No.
But from the previous one, that's what it's more clear. And yeah, that's the thing. So let us. Oh, sorry. Yeah, a question.
If you view gamma, the coupling as a joint distribution between mu and mu,
Yeah.
like, is there any intuition about what that represents?
In the unbalanced case, or in the. So what gamma represents?
If it were a joint distribution,
Yeah.
like a joint probability distribution, does it mean anything if you look at it that way, or?
Yeah. So the whole theory I've presented can be formulated in probabilistic terms. So in probabilistic terms, the problem of optimal transport looks like looking for a joint distribution of random variables, so that the expectation of the cost is minimized. So that would be the probabilistic formulation of optimal transport. And
You have two random variables with, you already know their distributions, but now you're looking at ways that you could define them jointly on a single probability space.
And we'll
But so that they each still have the right marginal distribution. And then there's
You can think of it as a transition also. If you start up in some place and you do like a Markov chain, just one step of Markov chain, what's the conditional distribution given where you started? That's what the transport plan.
is there another question before I move to the numerical parts? So I'll first describe how we write this problem down for it to be solved by a computer. So I will focus on the case where my two distributions are set of weighted particles, because this is the one that we can, that I will explain how to solve using algorithms. So this is my measure mu, my measure nu, and here I wrote what they are. They are sums of weighted points at locations xi's with weights pi's for mu, and similarly for nu. And if I rewrite the optimal transport problem with this new discrete setting, my cost function, it can be summarized by a matrix, which just says what is the cost of pairing the point number I to the point number j. And I will write a matrix of costs. So here is an example. I will, well, we don't really see the colors, but I used the yellow color to describe a small cost and a red color to describe a high cost. And then we have these two marginals, which are given by a set of weights that are represented at the end of the rows and the columns of my cost matrix. And what I want to do is to fill the entries of this matrix with mass. This will be my coupling gamma, such that when I sum over the column, I get the blue weights. When I sum over the rows, I get the red weights. And I want to favor yellow entries because these are the ones with the lower costs. So here is the solution that you'll get by solving this problem. And of course, it corresponds to an actual transport plan. If you come back to the problem with the geometry, we don't need to look at this problem this way. For the computer, it's just a cost matrix and two marginals. And this is a classical instance solving this problem of the so-called linear programming problem. So there have been methods to solve this kind of problem since the last 60 years. There are also dedicated methods in case you have special properties on your marginals. But these kind of methods, they do not scale very well to more than thousands of points. And we need to find methods to solve bigger problems more rapidly, maybe at the cost of doing a small approximation. And that's what I will describe. Here I've just shown an example. There are another class of methods which have emerged recently that can deal with interpolation of mass in low-dimensional space, so 2D, 3D, with very specific costs. But they do not solve our problem because they are very specific to this setting. But just so that you know, it's possible to compute optimal transport between, here it's two shapes which are discretized into millions of voxels, and we can solve it on a laptop in a few minutes. But that's not our aim is to solve for arbitrary costs in high dimension. And we will agree that we can do a small approximation of our optimal coupling to solve this problem rapidly. So here is the optimal coupling that I will get between these two distributions if I choose the square distance as a cost. And this is the product coupling that I've introduced at the beginning. We just does not care about the geometry. It just sends the mass everywhere. Maybe there is a class.
That's like making the random variables independent.
Exactly.
In the probabilistic version.
Yeah, in the probabilistic version, the product coupling corresponds to making your random variable independent. Yeah. And so what we look for is a family of problems which somehow interpolate between these two kinds of behavior. And this is what is done by the so-called entropic regularization of optimal transport. Instead of solving my initial optimal transport problem, which looked like this, I will add a small term, which is epsilon, a small real number, times h of gamma, which is minus the entropy of the coupling. It's just a function which tends to favor dispersed distributions. And the thing is that when I solve this problem, there are very efficient algorithms to solve this problem. So there are a few ways to interpret the introduction of the entropy in the model. It has been, for instance, a model introduced by Schrödinger to study gas. In economics also, it has been introduced, this variant of optimal transport, to deal with matching between agents with noise in their preferences. It can also be seen as a regularizer to stabilize the solutions of the optimal transport problem. Because here, with the optimal coupling, if I modify a little bit the positions of the particles or their mass, the solution can change a lot. But if I add this entropic regularization, you know that the solution will vary smoothly. And also it can be seen as a, yeah?
Sorry, maybe I'm anticipating what you already will show in the next slides, but you also show the unbalanced formulation where you go through the constraint.
Yeah.
Replaced by cross entropies. So is there a formulation with the entropic regularization and the penalty with cross entropies instead of the constraint?
So that's exactly my next slide. Not the next, but two next. So you're right. I will talk about how to solve variants of this problem. But first, let us see an algorithm to solve this problem. So optimal transport with a small entropy on the coupling. So first, I need you to accept a result because I cannot really explain where it comes from. It's a convex duality result. But the minimizer of the first problem, of the regularized problem, is known that it can be written under this form. So here, K is a matrix where I've taken my cost matrix and I've applied a transformation to each of its entries. I've taken exponential of minus the cost over epsilon. And then there is a theorem which tells me that my solution will be written as a rescaling of K. I will rescale the rows by some positive factors A's and the columns by positive factors B's. So if you accept that my solution of the previous problem will be written in this form, then we can come up with an algorithm. Because we know that the solution has to satisfy two marginal constraints. When I sum the columns, it must be equal to mu. When I sum the rows, it must be equal to nu. And so what I can do is just start with some choices of A's and B's. So it shouldn't be 0, it's 1, sorry. And then I can rescale the rows so that I satisfy the first marginal constraint. This will modify my weights A's. Then I can rescale the columns so that I satisfy the other marginal constraint. But then I will change the first marginals. So I need to repeat again. And it gives me an algorithm, which is called Sincon's algorithm. And it's not to converge at a quite fast rate to the optimal solution of the previous problem that I've written. And that's how it looks when you look at the evolution of this over the iterations. You start with a with a coupling which is not optimal at all, and it will deform the coupling until it reaches the optimal solution. So here I've explained.
Can you explain the images and what are the axes?
Oh yes, sorry, yeah. So this image, it takes the representation of couplings that I've shown in the first slide of couplings, where you have two one-dimensional marginals, and the coupling lies in the product space in between. And so here we computed optimal transport between mixtures of Gaussians, but the thing is that you will modify continuously over the iterations your coupling until you reach the good solution.
Why from such a concentrated?
Because at first you just send all the mass to your nearest neighbor. And then that nearest neighbor, in the next iteration, it will tell you, oh, you have too much mass coming to me. You need to redistribute somewhere, and you iterate like that. It can be interpreted as an auction with noise. Well, this I will leave you to figure out. There are many ways to interpret this kind of iterations. But here we have just solved the very standard optimal transport problem.
One thing that you might want to mention is that
yeah.
this is sort of a alternative minimization type of thing, and these a lot, but this one is already completely justified, and it goes all the way to convergence, and this is a completely kosher alternate optimization thing to do.
Yeah, exactly. So the convergence of this algorithm is very well studied and well known, and there are recent articles, by the way, with very good convergence results.
So this works for, you have to regularize to have this formulation.
Exactly.
But if you regularize even a little bit, then you get this.
Yeah. You get this algorithm if you regularize it.
And the solutions, instead of being concentrated on perfect paths, that it sort of fattened up a little bit.
Yeah, exactly this. The optimal transport problem without regularization, here because I've computed it between continuous marginals, it would have been exactly a map, a deterministic coupling, but the entropy smooths it a little bit. Yeah.
What did you say about this solution of the regularized problem? Will it continuously depend on epsilon?
Yeah. There's a continuous dependence on epsilon.
Epsilon tends to 0.
Yeah. And that's
regularized problem.
That's that's this animation. I've actually computed the optimal coupling with various values of epsilon, and in one limit you get the optimal coupling, in the other you get the product coupling. And so far we have just seen algorithm for solving standard optimal transport between two marginals which are known, but actually in many applications what we'd like to do is to include optimal transport in a pipeline where it's used as a regularization, or we want to solve maybe unbalanced optimal transport, we want to compute averages of shapes with respect to optimal transport distances, or 3D shapes in computer graphics. There are many more problems where optimal transport takes place. For instance, we can compute the evolution of a crowd. I won't explain how it works, but optimal transport is involved in that. And it turns out that all these problems, they can be solved if I generalize a little bit the minimization problem I want to solve by replacing the marginal constraints by functions of the marginals. So here in this formulation, if you replace F1 and F2 by functions which takes the value 0 if I satisfy my constraint, or plus infinity otherwise, then you just recover standard optimal transport. But actually, this formulation is quite general and allows to solve all these kinds of problems. And so I will just end on a generalization of the Sincon's algorithm that can solve this kind of problem. So you don't need to see the details, but it involves an operator which is called the proximal operator. It is a generalization of a gradient descent in a more stable fashion. And essentially, you can understand this variant algorithm as a descent on the rows and descent on the columns, alternating in the same way than synchron, but in a generalized way. And here again, we have convergence guarantees on this algorithm. And I've just ended up on showing a few convergence speeds for optimal transport problem. In the axis here is the number of iteration, and vertically it's the distance to the minimizer. Here I'm solving an optimal transport problem in blue, and in orange and red these are unbalanced optimal transport problems. So you have various convergence rates. They tend to be locally linear, and so it's possible to accelerate the rates of convergence. So in practice, you have to keep in mind that it's possible to go faster. And so that's the end of my talk. The main messages have been that optimal transport allows to lift a structure on the space to a structure on the space of distributions on that space. It can work, it can be solved approximately quite rapidly now with using these new methods for mixtures, for distributions which are several thousands of atoms, and also this theory extends to unnormalized measures. So I will leave you on this animation. Thank you.
Thanks everyone for coming. So I'm really excited to tell you guys about this work, our forthcoming work on learning developmental landscapes with my wonderful collaborators here at the Broad. And it uses techniques from optimal transport, which you're all now experts on. I'll do a little bit of review on that as well. So I'd like to start by showing you guys two images. So Conrad Waddington, a famous developmental biologist, produced a series of images to illustrate the epigenetic landscape, or the developmental landscape that a cell travels through on its journey from a stem cell to a differentiated cell. And his first image, or series of images, looked like a set of train tracks where a cell, switched between a number of terminal fates along its journey of development. But then later, his later image looks more like a landscape, and this is the image that people are more familiar with today. This image of a marble rolling down a developmental landscape. And kind of the difference between these two images is that in the image on the right, the marble is not restricted to any, finite set of paths. It can roll as it wishes along the trough. So in order to reconstruct this type of landscape, we want to be able to answer questions like, what are the origins and fates of cells at each stage of development? For example, at an intermediate position like this, the cell's origin, is some kind of undifferentiated cell at the top. And then at this intermediate position, it would have a limited number, but perhaps more than one fate available to it. And what are these fates? Okay, and also, what are the genetic regulatory programs that shape this landscape, and give it, the troughs in certain regions and the valleys in certain regions? So now, with the advent of single-cell RNA sequencing, we're able to actually measure this landscape. We're able to sample points. And here, I'm showing you guys a movie of stem cell reprogramming. So this is a two-dimensional visualization of a massive data set that we had with 65,000 cells. And each point is a single cell measurement. It's the measurement of the expression profile of a single cell. And the blue points are all cells from a single time point. So here, this is the first time point. And we reprogrammed cells over the course of 2 weeks. And over the course of 2 weeks, the cells moved through this landscape. And they moved to the left and then up, and they eventually branched. And this first main branch that we see, going off to the right, you can notice that a lot of cells actually disappear from this branch between these last two time points. And we ultimately found out, as I'll explain to you later, that this major branch, I can use the mouse cursor here, is cells actually dying along the way. And these are the successful cells at the top. And as you can see, this is not just a set of railroad tracks, right? This region here in particular isn't well described by, a tree with, a finite number of branches. It's more like the marbles that can roll along the path. And so this was our motivation, was to develop some kind of mathematical framework that can, encapture this. Yeah, there's a question.
So, as a two-dimensional plot, what are your dimensions there?
Oh, good question. So, the two dimensions come from a 20, first of all, so the cells are in 20,000 dimensions. That's the number of genes. But then we reduce it first to 20 dimensions using diffusion components, which is a nonlinear version of principal components. And then we create a nearest neighbor graph, and then we visualize that graph using a graph visualization tool called force layout embedding. Okay. So let me now tell you guys about our mathematical framework, which we call WADDINGTON-OT for optimal transport. So what we have here in this movie, what we had abstractly was a sequence of expression profiles collected at a discrete number of time points. And these profiles, as I said, these are vectors in gene expression space. And we model these expression profiles as samples from a time-varying probability distribution. So this distribution, PT, takes into account both the biological randomness of the underlying process, because the cells have some kind of stochastic expression profile, and also the measurement error, because we don't perfectly measure the expression profile of any individual cell. So our model is that we have samples from this distribution at a number of different time points here, and I'm just coloring each sample with a different color. Okay? So now, just to make a formal definition, we call this a developmental process. This is a time-varying distribution, not necessarily a probability distribution, because the number of cells can change over time. So this developmental process generalizes the concept of a stochastic process because it allows the cells to grow over time. So it doesn't necessarily have total mass 1 at each time point. Okay? And just, a technical note, I said we were sampling from this, but to sample from it, you first can imagine normalizing to get a probability distribution and then sampling from that probability distribution. So what is the most familiar stochastic process that people might think about is like a Markov chain. And a Markov chain has a transition matrix, okay? So if you're in some position at time t, where would you be at time t plus 1? This is the notion of a coupling between time points, which we heard about in the previous talk. But now we're thinking about coupling actually for stochastic processes or developmental processes, where we have a time series, and this temporal coupling tells you, conditional on where you are at this time step, where might you be at the next time step? Okay? So, for example, in this branching picture, if you were in this set of blue cells at the bottom at this time point, maybe you would be at this lower green subset of cells at the next time point. And these couplings kind of completely describe the branching structure, but in a totally non-parametric way. We haven't really made any assumptions about what the structure of the distribution looks like. So compared to the railroad tracks, we haven't really assumed that there's discrete branch points or one-dimensional paths or anything like that. And formally, you can think of this coupling as a joint distribution on pairs of points, telling where they would go at the next time point. Actually, mathematically, so this is kind of our key realization, is that this notion of a coupling for a Markov chain or stochastic process is the same as this notion of transport map that we heard about in the primer, and that I'll define in detail in a few slides. Now, in terms of these couplings, we can define rigorously the descendants of a subset of cells. So the descendants of this set of cells, for example, this lower green set of cells here, the descendants would be the cells at the next time point. Or you could also have the cells 2 time points later. And how do you compute the descendants, or how do you define them? Well, they're obtained by pushing a set of cells through the transport map. Okay, so if we take this set of cells and we come up with a vector with a bunch of 1s on these cells and 0s everywhere else, and then you just push this vector with matrix multiplication through the transport map, then you obtain a distribution on the cells at the next time point. Okay, and this distribution, like is indicated in this picture here, the descendants would be more concentrated in this, in this conceptual diagram on this group of cells here. Okay. Similarly, we can define the ancestors of a set of cells by pulling this set of cells back through the transport map, also with matrix multiplication, but on the other side. So here, if we had a finite data set, each row of this transition matrix, there would be a row for each point at one point in time, and a column for each point at the next time. So there would be a row for each cell at t2, and a column for each cell at time t3. So the row of this transport map would tell you where this cell at time t would go, and then by doing this matrix multiplication, you can figure out the ancestors of a set of cells S at the previous time. Okay?
I'm confused about the direction here. Like,
right.
If you're going forward, you said it's a matrix multiply, so I feel like going backwards should be an inverse. Is it?
So it's a matrix multiply, but on the other side of the matrix. Here we were multiplying on...
Oh, it's a transpose.
Yeah, you multiply a row on this side, and you get a row out. And here you multiply a column on the other side. Yeah, so it's transposed.
Wait, but what's this duality then? Like, how are they both rows and columns?
Okay, so here we're defining this for an abstract developmental process, but we can imagine if we had a set of cells, which we'll have in a minute, then for each row of the transport map, there would be a single cell at one point in time. So let's look at descendants because it's easier to think about going forward in time. So, okay, so for a single row of the transport map, that would tell you where a single cell at time t3 would go. For example, this individual cell might be transported over a distribution of cells that is concentrated here with high mass and has kind of low mass over here. So if we take a single row of this transport map, that's what that would tell you. It would tell you the distribution over where that one cell would go. And now given a set of cells, multiplying by this vector on the left will average those corresponding rows. So for all of the cells in this set S, you would average up all of those rows of the transport map. And that would give you one distribution over descendants. Okay? Yeah?
What if your sampling times misses intermediate states? So you're assuming that it's continuous with same speed of differentiation, but maybe between time 3 and time 4, there are multiple intermediate states that you're missing. So how can you account for that?
Right. So that's a good question. So here I haven't talked about estimating anything yet. This is just if we knew exactly what the transport was, then these would be the descendants of the cells. And then and you're right that if the time steps, so when we talk about estimating these transport maps, we'll only be able to do that if the time points are close enough. That's a good point. Yeah, question?
Sorry, but so you have more points in your picture, right, later than you have earlier.
Right.
I'm sure you're going to come to that. But then I'm just confused about what the matrix looks like. Is it the same size? Like, are you taking all of the cells that you're ever going to have that be in your matrix? And or what is how are you doing with birth and death and just in this picture?
Okay, that's a great question. So, so going back just to so if we have a developmental process, then the mass of this process at time 0 might be 1 if there was one cell. And then, for your own developmental process, if you have 20 trillion cells now, the mass would be 20 trillion. So the mass of the process is changing over time according to the total number of cells that there are in the organism that's developing.
But the number of columns wasn't the number of cells?
So here, you can think of it as a matrix. There's no rows. There's not really any rows or columns yet. But if you did have a finite number of cells, there would be rows and columns, and that would be the number of cells. And and so for a single row, the total mass of that row would be the total number of descendants that cell has. It would be the total mass of its descendants. So some cells might have descendants with, mass 0, which would mean they would die from time T3 to T4, for example.
But what phi actually is, just to make sure I understand, is that it's a coupling between two copies of expression space.
Yes. So we have a developmental process, which is a time-varying distribution, and the coupling, is a joint. It's a joint distribution, not necessarily joint probability distribution, but it couples the distribution at time, t1 and time t2. Okay. And now, so I'm just going to finish defining all these kind of population level quantities, and then we can talk about estimating them. Okay. So a trajectory, people talk about trajectories, but what is a trajectory? For us, the trajectory to a set of cells is the sequences of ancestors of that set of cells, and the trajectory from a set of cells is the sequence of descendants of this set of cells. Okay. And so now let's get to some actual data. If we are actually presented with data, we don't actually observe any of these couplings, right? So all we observe is some samples from these distributions at some time points. And we don't see the coupling at all, but we want to estimate the coupling. And after we estimate the couplings, we can try to infer the ancestors and descendants. Okay? So this is kind of the main goal of the talk. Okay?
Just to clarify, the ancestors and descendants are not discrete subsets of cells by the formulation you just gave necessarily.
Not necessarily.
They're distributions on the space of all.
That's right. Okay. So for any kind of real organism, these distributions would be discrete, right? Because you have a finite number of cells, I have a finite number of cells, but for some hypothetical, empirically they're discrete, right? Yeah. Okay.
But if you have some underlying model and there's biological variability, then
right, the true
thing might not be.
The true cells that John has are not determined when he's born because, it's not determined how much sun he'll be exposed to, so how much melanin is there going to be in his, if you go in the sun, you'll get more melanin, so this coordinate will increase, right? But it's not yet determined, so the descendants of your skin cells, 1 month from now, might be some kind of continuous distribution. But then when we infer the ancestors and descendants, those will be kind of putative ancestors and descendants that will be supported on the data that we actually have. And kind of the main assumption that we're going to make is that this process is moving in a particular way. So the transport map has a particular structure, or the couplings have a particular structure defining according to this classical mathematical technique called optimal transport. It's also sometimes called earth mover's distance because, as we learned in the primer, it was originally developed by a French mathematician to build fortifications and move piles of dirt. And this was used in Napoleon's army to build fortifications, because the British were coming and you had to build a castle as efficiently as possible. So yeah, how can we redistribute this red distribution to obtain the blue distribution? By moving all the piles of mass in some optimal way. And how do you specify how to move the mass? You do this with a transport map. So a transport map tells you how, when you take a column of dirt, how do you spread it over into a distribution of dirt, pi of x. So I'm using pi, Lénaïc used gamma. And now the optimal transport plan is specified by the solution to an optimization problem. Okay? So in particular, it minimizes the expected cost for some cost function. So for gene expression profiles, if you have some gene expression profile X and gene expression profile Y, there's some function C of xy, for example, squared Euclidean distance on gene expression space, that will tell you how costly it is to transport. And this is our strongest modeling assumption that we make. We're assuming that the distributions of cells are changing over time in a way that is some shortest path according to this optimal transport distance. Okay? And this assumption is kind of more and more reasonable, like your question, if the time scale is shorter and shorter. So assuming that these distributions are changing in some kind of shortest path, straight line sense is kind of reasonable over short time scales, but less reasonable over long time scales. So optimal transport, just to remind you, is a linear program, and there's very fast ways to solve it. So if you want to minimize the expected cost, you impose these constraints that the transport map has to, if you sum up over everywhere that you started from, you should get all the cells that you're going to. And if you sum up over everywhere that you could go to, you should get all the cells that you came from. Okay? So this is our stochastic process where we came from, the developmental process at time t1 and developmental process at time t2. And the basic idea is that we're going to be doing optimal transport from one time point to the next in order to estimate these couplings. So at a high level, this is what we're going to do. But we're going to see that we need to make some actual modifications to the standard formulation of optimal transport because, it was originally developed to move piles of dirt, and dirt doesn't grow, but cells can grow. So if cells actually proliferate at different rates, for example, you can see there's many more cells accumulating in this bottom branch. And this top set of branches is not kind of accumulating cells as rapidly. So this bottom set of branch is going to need to take cells from somewhere in the standard formulation of optimal transport. And so actually cells will kind of teleport from the top branch to the bottom branch. And this is not really reasonable, because it's kind of more reasonable to assume that cells don't just travel very long distances. We allow the cells to grow so that cells don't have to travel such long distances. So what we actually do is we introduce a growth rate function, and we modify the concept of optimal transport to allow cells to grow. Because if we see a cell with gene expression profile X, we actually know something about this cell. We know which genes it's expressing, because this is encoded in the gene expression profile. And we know if it's expressing some kind of cell cycle genes, it might grow at rate g of x. And if it grows at this rate for time delta t, then the total increase in mass will scale the mass of this cell by this factor. So if this growth rate is big or small, you'll get a big or small pile of descendants at the next time step. And what we have to do then is just include this in the right-hand side on the constraints here. So if you sum up for a single cell X, if it grows at this rate, then you scale the amount of mass coming from that cell. And what that means is if you just sum up over everywhere you could have gone, you get this rescaled amount. And then this is the last ingredient, so this is a little bit of a technical slide, but we use this unbalanced transport that Lénaïc presented. So we actually use this unbalanced transport here because we don't want to assume that we know this growth rate function exactly. All we assume is that we have some rough estimate for the growth rate, and then we don't want to impose these constraints exactly, so we relax these equality constraints to bounds on the KL divergence. And now what we're going to do is minimize and identify an optimal transport plan that minimizes this cost. So we're going to minimize the distance between cells at time t plus 1 and cells at time t. And we're going to identify a transport plan that minimizes this distance, subject to some constraints on this transport plan.
So that's your cost function that you put in?
Yes, this is the cost function that we use.
Is that super sensitive to the
normalization per gene?
No, we actually tried this with squared Euclidean distance in gene expression space and in diffusion component space and with a number of different pre-processing steps, and the gross features were all quite similar.
Well, I mean, even just Euclidean distance, the relative importance of each gene, right, is a normalization issue.
You mean if you do TPMs or log TPMs or something like that?
Yeah, sure. There's lots of ways, right? But I don't know.
Right, and we found that this wasn't hugely sensitive to stuff like that. Yeah?
I just wondered if you guys, I know in the past you dealt with this a little bit, but it seems like, so you kind of can estimate a potential growth rate based on the gene expression program, and I assume you could do the same for apoptosis or something like that. So when you have actual just cell division,
Right.
like in development, so you would get an increase in numbers of cells without necessarily seeing proliferation or, transcriptional signs of that necessarily. I just wonder, have you done this on that kind of data yet, or did you guys figure out a way to deal with that, or what do you think about that?
Yeah, you mentioned apoptosis, like cell death, and also cell growth. So what's important here is actually the relative growth. So if you have two subgroups of cells, and one is growing more rapidly than the other just in a relative sense, it could be because this bottom branch is growing or because the top branch is dying. And because we're just kind of sampling from this distribution, all we can know about is the relative growth. And so that's kind of the relevant parameter. And we just try to capture the relative growth, which combines all of these different factors, apoptosis and proliferation. Yes.
Sort of try to guess at that a little bit by looking at the actual programs you're seeing at a given time step?
That's right, yeah. So this growth rate function is, at this point, it's an input that we assume we have a rough estimate that we know ahead of time. And we don't derive it just from scratch from the data, but it's a function that you apply to the data that we kind of know beforehand.
And so then I guess it would be really hard to apply it in a situation in which you had cells dividing without necessarily a lot of proliferation, right? I'm just imagining.
Right, if they were dividing and you didn't know about why, you didn't have a good proliferative signature, then it would then you would be relying entirely on this unbalanced aspect, which can also capture growth. So if you ignore this G, this unbalanced aspect will still allow for growth. But if you have a very extreme rate of growth, then and you can capture that, then this will help. Inputting it will help. Right.
Yeah, so why have you done, it seems like you've overparameterized or something. I mean, why have you done both unbalanced and the G?
It's because we don't know the G exactly, but we do know something about the G, so it's helping a little bit. So this unbalanced transport doesn't know any biology, but by putting in the G, we're just saying we know a little bit about biology, so we're using that as best we can.
And you separately estimate G?
We don't separately estimate it. We just kind of guess it.
You guess G?
Yeah.
Each cell has its own growth rate.
Right.
And we believe the growth rate that we calculate based on the gene expression profile a lot more than the implied growth rate were we to sort of relax these constraints and allow the algorithm to figure out how much to grow and kill each cell.
But this is some function on all of gene expression space, so where does it come from?
We look at genes that are associated with cell cycle and genes that are associated with apoptosis and so on, and just assign a score that is a rate of growth based on if you have a lot of cycling genes, you get higher scores, and if you have a lot of definitely genes, you get lower scores.
So you have a bunch of key genes and you give them some positive and negative coefficients that sort of feel right?
Yes.
Okay.
Yeah, and we know roughly that one subgroup of cells doubles every so number of hours, and the other subgroup, roughly should double every, one subgroup might double every 12 hours, and the other subgroup might double every 48 hours. And
this thing is a base for an exponential, and you just have
we try to match that. Yeah. Yeah. Well, that depends on how tightly you fix these parameters here. So if you really crank on these divergence constraints. Like, for example, if you impose these exactly, then it'll be quite sensitive to this growth rate, right? But then if you relax these constraints entirely, then it will totally ignore the growth rate. And it will, if there's a cell that's hard to transport somewhere, it will just throw that cell away, you know. This is the entropy, so it's an entropic regularization term. Okay. And this makes sense both for algorithmic reasons and for kind of statistical reasons. So this this allows the transport map to spread cells out over more than just one descendant, for example. It allows the descendants to have more entropy, or it promotes that. And for algorithmic reasons, like we saw in the primer, this leads to very fast algorithms if you include some entropic regularization. And also because we don't know these distributions precisely, all we have are samples from them, you know, we don't need to match these constraints exactly. Yeah, but we did some numerical experiments just experimenting with different values of lambda and different growth rates, and we found the results were relatively stable to, you know, a range of choices for all these parameters. Okay. Any other questions? Yeah, sure. So we choose epsilon so that each cell transitions to a reasonable number of cells. So if you really encourage a lot of entropy in the transport map, then every cell will just transport uniformly to any other cell, which in the pile of dirt example would be like picking up a column of dirt and spreading it uniformly over everywhere you would want it to go. The other extreme example would be if you really don't want any entropy, then every cell would deterministically go to a single cell, which also seems kind of unreasonable. So we choose the entropy parameter so that each cell goes to maybe like 10 or 20% of cells at the next time point. You know, somewhere between 50% and 10% of cells at the next time point, which is
some probability. I mean,
yeah, right. That's the.
You're giving a cartoon of what's actually like a continuous distribution of tails, right?
Yes, that's right. So that's the percent of cells that have kind of a reasonable amount of probability.
Right.
Okay. Okay, so just to summarize, this is, by solving this optimization problem between every pair of time points, this is how we infer the couplings between time points. And this is how we infer where this developmental process is going and where cells came from. Okay, and we infer the ancestors of cells by just, you know, like I said before, taking a subset of cells and pushing it through the transport map. And we infer their descendants by also by matrix multiplication on the other side. So given a sequence of expression profiles, you know, we do this just like I said, and the inferred trajectory is the sequence of ancestors, and the inferred trajectory from a set of cells is its sequence of descendants. Okay, and so now let's see what happens in this reprogramming data set. Okay, so we have 10 time points with more than 65,000 cells total. And you know, this is our kind of empirical version of this developmental process. Okay? And when we compute these transport maps, we get the couplings between every pair of time points. And then given different subsets of cells, we can try to learn about where they came from and where they went. Okay? So here's kind of an overview of the landscape. So we can get a lot of this information just by looking at the gene expression signatures in different regions. So we can see that, you know, this region here was where a lot of cells were dying. And we call this the valley of stress because these cells were kind of stressed out and dying. And this group here, we call this the horn of transformation. So if you remember, we started off here on day 0 with mouse embryonic fibroblasts. And then by day 2, there's this kind of intriguing spike that we see. And from day 4 to day 8, the cells progress upwards in this kind of continuous fashion towards either the horn or the valley. And this is kind of the first major decision point that they make. And then after they make it to the horn, you know, if they end up in the valley, they're kind of stuck there. They don't really make it out. But if they make it to the horn, then they proliferate more and they end up either in alternative fates or at eventually, hopefully, as a mature induced pluripotent stem cell. Okay? And this is kind of a technical experimental detail, but we did this actually in two conditions. And the alternative fates that we saw actually depended on the medium that we gave the, when I say we, I mean, my collaborators gave the cells. So, for example, we see neurons if we give them serum condition after this, in the second phase of the study. So the way it works is you reprogram them by turning on all of these genes with a chemical called DOX. So imagine that they're in their little valley, happily in their valley as mouse embryonic fibroblasts, and you kick them out of this valley, and you try to reprogram them to become a stem cell, and you try to push them back up this hill. Okay? And after you kick them out of this hill, some of them become unhappy, and die, and they go over to this valley of stress. And I'm kind of waving my hands and saying we think they went like this, but the reason we know that is we actually looked at all of these trajectories to different tips of this landscape. So here.
Sorry, what can you just say one more time what we're actually looking at?
Okay, so this is called the force layout embedding of the data set. So this is a two-dimensional visualization of the 20,000 dimensional data. And each point represents a single cell gene expression profile. And these points are embedded by first computing the 20-dimensional diffusion components, which reduces from 20,000 to 20 dimensions. And then we reduce to 2 dimensions by visualizing the nearest neighbor graph in 20-dimensional diffusion component space. So this nearest neighbor graph captures which cells are close to which, and this 2-dimensional visualization is somehow capturing this as best as possible.
Right, and in this picture, you're not actually presenting anything from the transport yet.
Right.
You're showing this picture is purely based on the
expression. These look like neurons. These look like
and the
placenta.
colors are labels you have because you know when you collected each.
Exactly. This is, in some sense, the input data.
Right.
But we don't do it on 2 dimensions. We do the transport in 20,000 dimensions.
Right. So you went down to 20 dimensions with fusion maps.
Right.
And then?
And then for this visualization, we compute the force layout embedding, which you can think of as like TSNE, but it better captures the global structures. Okay? So here are the different trajectories that we see, and if you kind of piece these all together, you get the overall picture on the previous slide. But we're kind of decomposing this overall group of cells into trajectories to different tips at the final time point. So for example, the trajectory to induced pluripotent stem cells, which is this group of red cells here. So red cells are at day 16. This trajectory goes along the left border of the picture, okay, through this, what we call the horn of transformation. Whereas the trajectory to apoptotic cells passes through this broad region here and ends up in the valley. Okay, because we told it to end up in the valley, we asked what are the ancestors of this red group of cells, and the set of cells is at day 16, and we compute the ancestors at day 12, and then we compute the ancestors of those at day 11, the ancestors of those at day 10, etc., all the way back to day 0. Okay, and this gives us a trajectory to apoptotic cells, or senescent cells, or neural-like cells. Okay? And so these kind of trajectories are tracing out the different troughs and valleys in the landscape. And so why should we believe them? Well, we did a kind of a rudimentary check that they agree with what people actually saw in the past when they physically took a subset of cells and looked at the descendants experimentally. So you can take a subset of cells sorted out by FACS for different markers and then see how likely they are to go to become a stem cell, right? And this would be kind of what are the probability that the descendants of this cell end up as an iPS cell, and our kind of in silico experiments that we did just by identifying the transport map agree with the fact-sorted experimental data for this for these subgroups of cells. So maybe we could also believe them for other subgroups of cells. Okay, so now I'll highlight a few interesting biological observations that we made. So reprogramming success is predictable by day 2 because we see these 2 spikes at day 2. And one spike kind of has a high probability. If you look at the descendant distribution, the descendants are concentrated over here on the way to the valley. And this other spike has descendants that are concentrated here. Okay? Yeah, Sam, you have a question?
So this represents really all of the predictions that you made at some given point? I was just trying to understand how much of this...
So this is not all of the predictions we made. This is just for a few subgroups of cells that are either CD44 positive, ICAM negative.
It's not all negative in there.
Right?
So you're saying not all cells fit in each of those columns?
Well, this is also just one time point, for example, yeah.
Right, so I'm just trying to understand, out of the cells even from that time point, what does this represent in terms of the percentage of predictions you made?
Yeah, I guess these are all of the 6 let's see. So
you're missing all positives, but maybe those don't exist.
Yeah, so yeah, right. So this is not all the possibilities. So
maybe that's
yeah, I mean, there would be some cells that are all positive as well. But for these 6, which they measured experimentally.
And how big a portion did these, okay. Like, is this like a lot of what you did at that time point? Is this?
This is, we can take this offline. I think this is a lot of, this is not just a small subset of, this is not like 3 cells. It's like a representative sample somehow.
Okay.
Okay, so we can look at the descendants and see where they go. We can also look at the ancestors and see where they came from. Also, so now we're going to look at the ancestors all the way back to day 2 of groups of cells that were on day 16. Okay, so we're going from the end all the way back to day 2. Here we were going from day 2 forward to day 8. But now if we go from day 16 back to day 2, we can look at the enrichment in these 2 clusters of various day 16 subgroups. So for example, if you come from an induced pluripotent stem cell, then your ancestors will largely be in cluster 4 and less likely in the other clusters. So this is just kind of exploring where cells came from and where cells went. Okay, and these, this split that we see on day 2 is somehow predictive of reprogramming success.
So all this relies on choosing epsilon so that you have some amount of determinacy, right? And otherwise you
right.
tell stories like this.
Yeah, right. Then every cell would just go everywhere if that was the case. So just to wrap up in my last few minutes, I'll tell you guys about kind of one cool thing that we did. Another cool thing that we did was to learn gene regulatory networks. So the picture that you can have in your mind here is that there's some kind of vector field, and the cells are flowing along down this vector field, and we want to learn this vector field that summarizes the transport probabilities. So what we can do is we can try to learn a differential equation, x dot equals f of x, such that when we look at the pairs of points that we get, pairs of points at time t1 and t2, we actually match the transport probabilities. So we want to find a function f that specifies a differential equation such that when you push a point at time t1 through f, it matches what you would get according to the transport probabilities. Okay, and when we do this, what comes out is some kind of list of transcription factors that kind of show where the cells are flowing and why they're flowing that way. And just to tell you the conclusion.
What comes out? Like, if f is linear, then the coefficients tend to be concentrated on some set?
Yes, that's how we did it. Yeah. So we, there's a lot of details in how we specified f.
Sure.
We found we looked for a low rank linear f.
Yeah.
Okay. Yeah.
Okay, so linear and then L1 regularization.
Yes.
And then you got
And we got a list of transcription factors that drove the cells to different directions. And if you look at the transcription factors that are driving cells along the successful trajectory, one of the strongest genes that's pushing you the most in that direction was called OBOC6, which wasn't very well studied before, but if you actually force the expression of OBOC6, it actually increases the probability of successfully reprogramming. And so this is something that came out of the model. So this is cool. So if you look at this bar graph, this is just showing that without forcing the expression of OBOC6, there's some efficiency and it goes up. And this is another gene that we kind of knew would do this. So beforehand.
People didn't know this stuff.
People didn't know about OBOC6 before. Yeah? Okay, so that's basically it. I'll leave you this picture. There's a lot of stuff in the data that I didn't have time to talk about. Like we saw, some interesting copy number variation stuff going on in the valley of stress. We also saw that the cells on their way to becoming mature iPSCs, some of them became placental-like cells, and there was interesting cell signaling from the placental cells to the iPSCs. Also, on their way to becoming iPSCs, they reactivated their X chromosome because they happened to be female cells. So they had 2 copies of the X chromosome. So there's a lot of cool things in the data, but I wanted to just kind of introduce you guys to this, this mathematical framework for identifying trajectories. So, so thank you for your attention, and there's time for questions. And I'll thank my wonderful collaborators.
So there's plenty of time if you want to go through your last couple of slides. Like,
okay.
We usually go right to 11. So.
Okay, great. So I can. Okay, so I can tell you about one more thing, since there's a few more minutes.
Yeah.
So, okay, great. So what I described to you so far was a method to identify the transports from kind of one time point to the next, okay? But one interesting mathematical direction, at least, to extend this model is what can we do if we have a lot of time points, okay? So if we have a certain amount of data, and this data is spread over many, many more time points, first of all, why would we want to do this? Maybe we want to estimate these differential equations, and this is easier to do if you have a lot more time points, right? Because we're trying to estimate a derivative. Then this simple optimal transport procedure that works from one time point to the next would do something kind of silly. It's just going to say, oh, we go straight from this. If you think of this as kind of a one-dimensional regression problem, this is some kind of regression problem where each of these data points is an empirical distribution that we saw. And the optimal transport will go directly from one distribution to the next. And it would be kind of like doing a regression where you just connect all of the dots. But instead, we want to do something smoother, because if you don't have a lot of data at each time point, then you can think of these dots as being quite noisy. Okay? So we want to do some kind of distribution-valued regression in order to interpolate between the time points. Okay? And this is kind of an open question, kind of algorithmically, what's the best way to do this? So one idea that we have that's kind of partially formulated is to do something with Wasserstein barycenters. So if you have a one-dimensional regression problem, a simple way to identify, if you want to find a function f so that yi equals f of x, okay, so you want to express the y coordinate as a function of the x coordinate, what you can do is just average all of the y values near a desired x. So if you want to interpolate the function at this point, you can average all the nearby dots. And this average is kind of like finding the center of mass, okay? So now how can we do this for distributions? Well, the center of mass for distributions, as was defined in the primer, this center of mass for distributions is called the Wasserstein barycenter, and it's the distribution mu that minimizes the optimal transport distance to all of the nearby data points, okay?
With given weights?
Yeah, so we can imagine that these weights are just all of the, so if you want to estimate this distribution at time t, then you could look at all of the distributions that are, for example, within some interval around t. And then you could find the barycenter of all those distributions.
So for two things, this is the standard interpolation, but for more, is it like a well-defined, well-studied object, or no?
Yeah, this is the Wasserstein barycenter for, so for 2, it's just optimal transport, right? And for more, it's the barycenter of the distributions. But, this is an infinite dimensional optimization problem. You can discretize it. But to motivate this in another way, imagine that at one of our time points, we have just one cell. Suppose we take this nice single cell data set and someone gives me more data, which should make things better, but they give me an additional time point with only one cell. Then if I do optimal transport, into this time point, then everything will be transported to this one cell, which is kind of unreasonable, right? So we want some way in which we can kind of smooth over time and just have the method get better as you add more data to it. So right now we would just have to ignore that time point, for example. Yeah, and there's a lot of kind of other data sets that we could apply this to, that we're excited to apply it to. In general, data sets where there's a time series, right? Because we need this time information. There's also, interesting experimental design questions, like how can you select optimal additional time points, for example. And we're also interested in thinking about doing intervention studies with CRISPR knockouts, and also incorporating cell-cell interactions in these models for learning gene regulatory networks. And there was a lot of cool cell-cell interaction stuff in the data set as well, that we can talk about offline as well. So I guess with that, I'll take some more questions. So thank you for your attention. Oh yeah, question?
I can imagine that the method would be somewhat sensitive to non-uniformity of sampling from all cells. But do we have some sort of a robustness guarantee against non-uniform sampling and that sort of thing?
So yeah, you're right. If you're not sampling from the actual, like if there's some true developmental process and you're sampling from a different distribution, for example, because some cells are sticky or something like that, and you don't collect them, then you'll be sampling from some other distribution, and it's kind of hopeless. But in terms of and also, we haven't really proven anything about this either, so there's not really any analytical guarantees, but that's an interesting area for future work as well. But kind of as you get more samples, we expect our empirical estimates, and this is obviously well understood, is how these empirical versions will converge to the true version as long as there's no bias in the sampling. And then the transport maps will also concentrate somehow, and people understand these pieces kind of individually, but we don't really have guarantees for the whole thing as one pipeline, I guess. Yeah?
So you're impairing these transport probabilities, but in your map you have, for the same time point, multiple fates. How do you extract those fates from this probability distribution? That is to say, in time t, you can go to multiple different stable points.
Right. So yeah, a branching event is somehow when the fates become bimodal. So if you have kind of two main areas where you could go, this would be some kind of branching event. And this is encoded in the transport map. But we don't really extract it explicitly at this point.
All right, there's no more questions right now. Let's thank Jeff again.